Hey, my BIOS 115 students, how are you today? It's the last lab that you will have to perform for this class. And in my opinion, it's one of the coolest ones. In fact, you know what? I'm just a turtle ecologist, right? That is my field of specialty. But I love this stuff. I think this stuff is so totally cool. That is DNA sequencing, electrophoresis, and it's just really neat. And I hope you enjoy this simulation. We do this as a wet lab too, and it's all sorts of fun. And I will say that this virtual version of it, they do a really nice job simulating what actually happens in the lab, which is why you see an empty lab bench, and I haven't brought anything out yet because it is like a step-by-step sort of occurrence as far as getting everything. They speed it up for time. It's usually about a 3-hour lab. Obviously, it's not going to be that long. All right, so you read about DNA, and you printed this lab out. It's 8 pages. Oh my goodness, but there's a lot of procedure involved here and also a lot of background. As you know, DNA is the blueprint, what makes you, you. And we've come a long way in the last 50 years as far as DNA technology. And one of those technologies, I'll let you look through that, not only on this lab handout, but also in your readings, your textbook readings, that a gel electrophoresis can separate sequences of genes, can separate sequences of genes and map them out. I know I'm being terribly simplistic with this explanation. It's more complicated than that, but for our purposes, you can read about that, but just to accomplish this lab. And I'll get more into the explanation, like kind of as we go, as needed. Those DNA samples separate out into bands of DNA fragments. And you'll see on your lab handout, page 4, there's an example of that. And those whodunit types of shows will actually sometimes show these gel electrophoresis results to see if somebody is the father of a baby, if their DNA matches, or if somebody is actually, like if you have 3 suspects at a crime scene and they collect DNA samples, which one was the one who was present at the crime scene and who committed the crime. So since this is a little bit of a involved technique, we'll go ahead and get started. So you're going to run this gel electrophoresis, and what we're doing is ultimately we are going to find out who is the father of this child. We have the mom's DNA, we have the child's DNA, and 2 people who could be the dads. And we're going to determine who's the daddy. So we are going to take an electrophoresis chamber from the instrument shelf and place it on the workbench. And then we are going to take a gel and place it on the electrophoresis chamber. And then we're going to add 250 milliliters. Oh, somebody's home. Sorry, I'll keep talking through this. Then we'll add some buffer solution. So let's do that. So here's our electrophoresis chamber. We're going to put that down, and that is very close to what they look like, which is totally cool. They do such a good job with this. This is the electrophoresis gel. Now, if you were taking this class on campus, you would have to make, you'd have to make your own electrophoresis gel. It's basically, when you heat it up, it's a liquid, and you put it on one of these molds, and it's like the consistency of firm jello. That's where your DNA samples are going to run. And if you look closely, whoop, not that way, you can see these are the wells. They're little divots in the DNA or in the gel itself where you put the samples of DNA. And then when you'll see, we'll plug that in and you'll see how they separate out. Hey, Betty, shush. Trying to do a video here, kiddo. It's my last one. Okay, so then what we're going to add is we're going to add the buffer, 250 milliliters of the buffer solution. Okay, let's see how we can, I can't see what that is. It's behind my bar. There it is. That is it. I kind of thought that's what it was, but I wasn't sure. So we're going to go back and we're going to take our buffer solution and we are going to add it to the electrophoresis chamber. So let's do that. I have to zoom out a bit. There we are. So we're going to add that and we're going to put in 250. And now this is something that you would actually do in the lab as well. It's a liquid and it can be a little challenging because you have to put enough on so it covers that slab of gel. And sometimes if you don't pour the initial gel right, it just floats around or disintegrates. It can be very challenging. Obviously, that's not going to happen here because this is a virtual simulation. Okay, next. So we take these microtubes and a micropipette from the shelf and we're going to label them 1 through 5. That's where we're going to put our DNA fragments. So we're going to go ahead and take those and we're going to put them on the shelf, label them 1 through 5. Okay, there we go. Here's our micropipette. We'll take that down too because we're going to use that. Whoop. Come on, need to label you guys. Let me label you. Darn it. Okay, there we go. 1, 2, come on, 3, good, 4, and 5. Okay, we have done that. Take the 100 BP DNA ladder and the following Vinter fragments from the shelf. So the DNA ladder is what we compare this to, and it's for measuring sake. Then we're going to take the DNA from the shelf. And we'll place them on the workbench. And then we're going to add the reagents into each of the microtubules. Okay, so let's see what we have here. That's the 1 kp ladder, the 1 VP ladder. Okay. Okay. How much are we putting in? I should have checked. 50 micro. Okay, 50. Okay, so let's put 50. There we go. Okay. And there we go. Okay. Oh, wait. So that's in number 1. Number 2 is the DNA from the mother. No. No. There we go. Okay. Where'd our micropipette go? Oh. Little tricky. Okay. So 50 micro. Done. We're going to dispense that in number 2. All right. Good. And this goes there. And this goes here. Now, okay. The next one is the child. Okay. Here we are. And that's for number 3. Good. Okay. And number 4, the fourth one, let's take that down, is going to be, there's the mother, the child, and possible father number 1. Whoops. Good. Okay. Done. Good. Okay. And possible father number 2 goes into the last one, into number 5. Draw 50. It's done. Okay, great. Let's clean up. Good. Good. And good. I think we're going to need that back. Okay. Take the 2X gel loading buffer from the container shelf and place it on the workbench. Using the micropipette, add 50 microliters of the 2X gel loading buffer to each of the microtubules. Okay, let's do that. So 50 in each. Let's take this down. Did I pass it? There it is. Okay. There we go. And I'm glad they make it this color so we can kind of keep track. Good. All right. Okay. Done with that. And clean up. And there we go. All right, set up the electrophoresis chamber according to the directions in the table for each sample being tested. Move the micropipette into the microtube, draw 5 microliters, move the micropipette into the electrophoresis chamber to insert the DNA sample. Select the lane specified in the table. Okay, and then we empty the DNA samples, and then we clean up. Okay, because it doesn't let us go on until we clean up. Now again, I love how closely this simulates what we do in the lab, because then this is the next step, is we have to load the gel, it's called, and you see these chambers here, is we put a little bit of the sample of each into those little wells, into those little holes in the gel. And remember, the buffer solution is floating, like this is submerged under buffer solution, and to do this carefully, under the buffer solution to get the liquid sample in. If you do it wrong, it just like floats away. Sometimes people will like stab the gel, the electrophoresis gel, with the tip of the pipette, the micropipette. I mean, it's just, it's definitely a technique that is tough to master. I'm kind of glad that they, all right, so anyway, so here's the first one. We're going to draw up 5. And then we're going to where's the pop-up? Okay, dang it. All right, hold on. Oh, dang. Where's the pipe? Set up, okay. Move the micropipette into the microtube. Drop. Move the micro onto the electrotube to insert the DNA sample. Select the thing. Okay. Yeah, that's what I did. Come on. I did this before. Again, this is like the. No, come on. No. Why is it not? Come on, come on, come on, come on. Did I not draw it up? Ah, come on. Oh no. Okay, let's try again. Maybe it's gone a little wonky. Okay, so I drew that up. Where's my pop-up window? Oh no, why are we not working? Ah, that's why, because I would. Oh jeez. Okay. Okay. All right. Done. This goes into lane 2. Okay. I'm glad that worked out. Lane 3. Okay. All right. Great. Okay. So now we are. We're done with these. Excellent. Very good. And now what we do, now that all we've loaded the gel, that's what that's called. We put samples of, we put the samples into each of those wells or each of those lanes, right? You'll see why they're called lanes. Press the up arrow of the chamber's power supply to run the voltage at 100 volts. This is why it's called electrophoresis. Then we're going to press on and start a timer or note, and it runs for 1 minute. So it's called electrophoresis because this is an electrical current. You see like there's negative on one end and positive on the other, like there's red and blue. And here are your DNA samples, and it runs an electrical current through the gel, and that moves that sample, and those fragments get laid down according to their size, depending how far they migrate, which is what gives us the banding. Appearance. So at 1550, we'll go ahead and we'll turn the gel off. If you don't run it long enough, you won't get the banding. If you run it too long, the bands wind up falling off the gel. I've seen that happen. And the other thing is, when we do this, you can buy these simulation kits where they have fake DNA. We have the electrophoresis chambers at school. We have everything, but we just don't have DNA samples. But what has been discovered pretty long ago is that the dye that's used to make Kool-Aid, you know, that drink that's so terrible for you, actually, the different colors, if you load them up like DNA, they'll band out just like DNA. Like orange Kool-Aid bands out into red and yellow. And then blue Kool-Aid, which I think is what raspberry flavor, that bands out into the green and yellow. It's actually, it works well, and it's so cheap to do, right? Okay, so now we've turned off our electrophoresis unit. This can take up to like 30 minutes, an hour in the lab. Okay, so then what we do is, now when we in the lab, we don't need to do this step because we use Kool-Aid colors, but if this were real DNA, what we do is we have to soak this in, oh, what's that called? I'll come across it. To make because the bands usually aren't visible, you actually have to soak it in something bromide. Oh, I'm not looking. Anyway, and anyway, we're going to do this. Take the UV viewer from the instrument, place it on the workbench. Oh, erythidium bromide, right, which is a carcinogen, which is kind of tricky. We don't use that at NAC. I've used it at other places when I've done this lab. But all right, so we're going to take our UV box and put it on here, and we're assuming we've already soaked it in the erythidium bromide. We're going to there we are, and see how it is banded out. How cool is that? So we can see those bands, and we kind of pan around, and this is our reference. That's not an actual sample that we sampled. Remember, here's lane 1, or it's actually lane 2, but this is lane 1 with the ladder sample. Here's the mom, and her DNA is here in 2 bands, right? This is where it began, and then they separated out. Here's the child. See, they have this band in common, but here's one that the mom doesn't have. Here's what they think. This guy might be the dad, or this guy might be the dad. So the mom and the child share this band. Now, who does the child share this band with? This guy or that guy? Oh, okay. And what you can do is, here, measure this. What it wants you to do is, we measure the distance from the wells to the bright leading edge band using the metric ruler. And you want to record that distance in step number 18. You see 18, right? Where is oh wait, hold on, where'd the ruler go? Oh, no. I did this before. I might actually not have you do that if I can't find it. Where's the ruler? It's got to be here somewhere. At the left of the UV screen that slides up and down when you drag it. Okay. Oh, shoot. That's not what I wanted to do. No, I didn't want to do that either. There we go. Okay, so you're going to, for 18, hold on. Yes, for 19, you're going to use, in 19, it shows you how to use a ruler, but for 18, what you want to do is you want to write these steps down. Hold on. So here's that first, write that measure in, and then that one. Do you see that? I think my cursor's in the way. Let's go back to the beginning. I'm sorry, and you can pause the video. So there's that one, the first one. Then we have this, right? There. So write these down. You see the numbers as they go by. There we go. And there. And there. And there. And there. There you go. Now, as far as table 1, you want to write down the distance traveled by the leading band. Okay. Hold on a second. Let me get the ruler back out. Okay, so the leading band here, that's the leading band there. Write that down. There's a leading band here. There's a leading, or, I should be more specific. Here's the leading band for this lane 2, the mother. Here's the leading band for the child, right there. See here? Here's a leading band for the first guy. She thought could have been the dad. And here's the leading band for the second guy, who she thought could have been the dad. So you want to put that in table 1. Those are the leading bands. Oh, it has like the lanes, okay. And then, last but not least, under table 1, with all of this data, which is the dad? Was it the first guy or the second guy? Oh boy, drama, drama, drama. Okay, so that is that lab. Very, very cool. It is unfortunate you couldn't do this in the laboratory because it is a really neat exercise, but nonetheless, here we are. And it's virtual, and that's cool too. All right, everybody, I hope you enjoy the rest of your day, and we will see you soon. Take care, guys. Bye.